Human genome sequencing.
DNA sequencing is the process of determining the exact order of the 3 billion chemical building blocks that make up the DNA of the 24 different human chromosomes. This information has helped reveal over 20,000 human genes within our DNA. Here's Jarrett with some more fascinating information about the DNA in our bodies.
Enzymes work by lowering the activation energy of a specific reaction. This, in turn, increases the rates of both the forward and reverse reactions. The reactants, or substrate when referring to enzymes, bind to the active site in the enzyme, which brings them close enough together for them to proceed with their reaction. DNA is made of 3 components. A nitrogenous base, either adenine, guanine, cytosine, or thymine, is attached to the 2' carbon of a 5-carbon sugar known as deoxyribose. The phosphate groups of DNA form the backbone. They are attached to the 5' carbon of one nucleotide and the 3' carbon of the next nucleotide. The bond that joins the nucleotides is called a phosphodiester linkage. DNA is a double strand, but one of the strands runs in the 5' to 3' direction, while the other strand runs in the 3' to 5' direction. They are also said to be complementary with their nucleotides. That is, wherever one strand contains an adenine, the opposite strand will contain a thymine. Likewise, wherever one strand contains a guanine, the opposite strand will contain a cytosine. In a study conducted by Morton Rasmussen and company, they successfully sequenced the genome of an approximately 4,000-year-old human using a tuft of hair that was preserved by permafrost in the Kekkatorssuaq region of Greenland. Their data is authentic due to multiple analytical techniques. The first one is known as single nucleotide polymorphism analysis. An SNP is simply a difference in a single base pair of DNA. These are the most likely to survive through time than any other form of genetic variation. SNP analysis of the permafrozen hair indicated contamination levels of less than 0.8%. Haplotypes, or groups of alleles that are generally transmitted together, from mitochondrial DNA and Y chromosome DNA are the same or very similar to those of Northeast Asians. Admixture analysis, which use SNP data to estimate the most likely ancestry, do not show any European components. Finally, PCA plots show close similarities between the Kekkatorssuaq genome and current Northeastern Asian populations.
Single nucleotide polymorphisms, or SNPs, are simply sequence variations between individuals at a particular point in the genome. Each individual has a million or more of these SNPs. SNPs have shown to be responsible for differences in genetic traits, susceptibility to disease, and response to drug therapies. SNP analysis is reliable in determining variations between samples. This analysis is recommended in cases in which samples have been subjected to extreme environmental conditions such as extreme temperatures, as is the sample used in the article. Another form of DNA analysis is in the use of mitochondrial DNA, often referred to as mtDNA, and is present in the mitochondria of every cell in the human body. mtDNA analysis is especially useful for samples which are degraded, exposed to harsh temperatures, or of small quantities such as the tuft of hair used in the article. While mtDNA analysis is not only easier than other forms of genetic testing, mtDNA has a fairly steady rate of mutation that permits a reasonably accurate estimate of exactly when a particular group of people migrated from their primary group. Thus, the mtDNA from the remains of a deceased person can be tested to determine the specific racial group from which the individual came, and in addition, the approximate time when that individual's ancestors migrated from their primary racial group can also be determined.
In the case study, the first piece of information that needed to be determined was how old the specimen was. This was calculated using the ratio of carbon to nitrogen of the ancient hair samples and of present-day samples, and also ancient reindeer bones that the age had already been determined for. Using this data, it was discovered that the hair of the human in question was approximately 4,000 years old. The following figures show the results from the 3.5 billion chromosomes that were read from the hair strands. Of these, researchers were only highly confident of the validity of approximately 350,000 of them. Allele repetition results in the chromosomes being easier to read and the data being more thorough, but if there is a duplicate allele, a portion of the chromosome cannot be used in the data because the duplicate is an error that occurs when a gene is missing. This is why the researchers were only confident in a small portion of the chromosome readings. These chromosomes were also the ones that are used in the further analysis. The final images show the distribution and lineage of the accurate chromosomes. The most important figure as it relates to the question of the experiment is the PCA plot in the upper right-hand corner. A PCA plot is frequently used in DNA sequencing, and it shows how related two individuals are in terms of genes. This PCA plot is slightly different as it groups together the average DNA sequence of humans from different parts of the world so that the average DNA sequence of the macaque can be compared to them. The purpose of this is to roughly determine where the macaque migrated from. It was hypothesized that the macaque moved in typical native patterns from Western Europe to Greenland, but this is not the case. The PCA plot shows that the macaque are most closely related to Eastern Asians, and so they migrated from Eastern Asia through North America to Greenland instead.
Whole genome sequencing has also been implemented for hundreds of animal species. In this supplemental article, Whole Genome Sequencing, an analysis of the Malaysian cynomolgus macaque genome was completed using a resequencing method based on the Indian rhesus macaque genome. Approximately 9.7 million single nucleotide variants and SNVs were found between the Malaysia cynomolgus and the Indian rhesus macaque genomes. Analysis of this data revealed that the Malaysian cynomolgus macaques have experienced at least 3 population bottlenecks. This figure contains an overview of the Malaysian cynomolgus macaque genome sequencing and analysis. This figure displays the SNV discovery rate and rhesus macaque genome quality. The red line represents the rate of homozygous SNVs, and the blue line represents the rate of heterozygous SNVs. SNV sites with quality values less than 45 were filtered out of the sequencing data for improved accuracy. This figure shows the number and length of indels found using the information from high-quality SNV reads. The term indel describes a special mutation class that includes both insertions and deletions of nucleotides in certain regions of the genome. This final figure shows screenshots of the macaque genome database, which were now analyzed for phenotypic diversity among the different species of macaques.
In conclusion, whole genomic sequencing has a number of useful applications. It can be used in the context of paleogenomics, as demonstrated by the 2 articles cited in this presentation. It can be used to detect important genetic differences and disease susceptibility of individuals of different ethnic backgrounds. And, most importantly, for the human race, it can be used to develop preventative and personalized medicine.